going to be hard to finish, so I'm not going to worry if I don't finish, but it's good. And we did, okay, here we go. Now, chromosomes contain DNA. DNA contains genes. So from bigger to smaller, chromosome has everything. Chromosomes have DNA, and DNA has your genes. So that's the order of things. Proteins, workhorse is good. Proteins do everything you need to do. I mean, they make muscles, they make enzymes, they make hormones, they make your hair, they make the color. Proteins are your building blocks. And every protein you have is coded for by the DNA of the nucleus. But he can't leave, so he has the message goes to the messenger. I just said enzymes are proteins. Proteins break down the food you eat, so you got to have them for that reason. You need protein to make new cell parts, so you cut yourself and you heal. Enzymes like the polymerase is in charge of replication and making new DNA. So you just can't. You got to have proteins. Proteins are what you got to have. And we've been discussing all this stuff just because it revolves around proteins. We don't have a lesson like this about fats. We don't have one about carbohydrates, because carbohydrates is your gasoline. You don't deal with them. Fats you could actually do with less, but the fat is part of your cell membrane. But the other part of the membrane are proteins. All right? When you express a gene, you make a protein, because that gene determines what kind of protein you have. For example, your blood is red because of a protein called hemoglobin. Well, there's a gene that makes that hemoglobin. You may have A type, or B type, or AB type, or O type, but the gene determines what you have. And the transcription, by now you should know, that's how the RNA messenger is made. And the translation happens outside there in the cytoplasm. And that's when the ribosome actually makes the protein. So in the nucleus, the DNA unzips, and one side is used to make the messenger RNA. That is transcription. That then leaves the nucleus and goes out into the cell and finds himself a ribosome. And the ribosome, like Braille, reads that messenger and makes the protein. That's translation. You translate what you've been given. Now, on the messenger, there are a number of things that you'd be aware of. And there's a three-step process: initiation, elongation, termination. Initiation has to start the process of translation. Now, we're talking about at the ribosome. We're inside the plasma. So something must start it, and something must keep it going, and then something must stop it when it's over, or it's been run on forever. So let's talk about it. You know how to read the mRNA. You know that if you see an A, you come back with a U. If you see a C, you come back with a G. If you see a G, you come back with a... You know all that stuff. So that part is no problem. But did you know that every three letters mean something? There's not one. Every three letters mean something. So when I give you examples, I try to give you 3 or 6 or 9 or 12. I don't want to do 5, because 5 is one shorter than 6. So these three bases might code for one kind of amino acid, and then those three might code for another kind. This is a codon, and every three is a codon. So when I say, here's a codon, A, and I now say, give me the anticodon in RNA, and you would say U. I would say, here's a codon, C. Give me the anticodon, and you would say G. Because you got your three letters. Okay. If I said this codon only contains C's, write for me the anticodon. You can't say C, can you? You don't want to say G, 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 G. You want to say there's 3 of them. So you know what you're talking about. All right? So codon, the amino acid. Proteins are made of, words are made of letters. Well, proteins are made of amino acids. And if you take a word and swap 2 letters, you change the meaning of it, do you not? Same thing with the protein. If you swap enzymes, then that's a whole new word, a whole new protein. Now, the order of the protein is determined by the DNA. Because he's going to tell you the order to put those letters in to spell that word. But if it's the messenger who actually goes outside and tells the ribosome, here's what this guy wants. You got to do it for me. Because he told me this, and I'm telling you this. Okay? Now, you know about a eukaryote has a nucleus. It has all these membrane-bound things inside. It's a very complex-looking cell. And in a eukaryote, as you know by now, the messenger must leave the nucleus and go out into the cytoplasm, find himself a ribosome, and get it done. Well, what if you have no nucleus? What if you don't have a nucleus? It's even easier. You just unzip the DNA, make your messenger, bam, right there's a ribosome. That's simpler, is it not? We know are simpler than a eukaryote. So in the prokaryote, there's no nucleus, so naked DNA, which means not inside the nucleus. Now, the question is, well, if there's nothing there stopping the DNA, why can't DNA talk to the ribosome directly? What's your problem? It's built on reversing. If I'm the DNA, you're the ribosome, and I say up, what's the opposite you're going to say?
Down.
I don't want that. I want up. So I got to still use messenger. I'll say up, Katrina will say down, and you do up. So even in a prokaryote where I could talk to you, you're going to get me totally backwards. So I still got to use my messenger. Even in a prokaryote, she ain't got to go anywhere. She leaves me to go right to you. But I'm still talking to the messenger. And basically, as the messenger is being transcribed, she's going right into a ribosome at the same time. It's like everything happened at one time. But still, if you're the ribosome, I can't talk to you. Because you're going to do what I say, you're going to do the opposite. That's going to mess everything up. So I still got to talk to my messenger. And then she got to talk to you, get it right. And that's how that works. Now, there's got to be something on the messenger that will plug into the ribosome to start the process. It can't plug in halfway down the messenger. It has to start at the top of it. That's called the pre-initiation complex. Now, these factors, 1F1, 1F2, 1F, I don't know what they mean. But I know that they're on the end of the messenger who inserts himself into that. Are you anybody diabetic beside me? When I take my blood and I stick my meter, that meter comes on by itself. I don't turn it on. It just on. If I put it on backwards, that's going to happen. So I'm thinking that just like my little blood tab, when I stick it in there and it activates this thing here, when this proper end goes into the ribosome fires up, ready to go. And that's why you need the initiator complex. And then the 1F, I never did find out what the F stood for. I guess it means it's right here, initiation factor. Could that be too simple? Sometimes you think, ain't that simple? But I think it is this time. I think it is that time. I think it really is this time. All right? Now, you do recall that a ribosome's got a big subunit and a small one. Do you know that? Right? It's got a big piece and a little piece. Okay? This is the first amino acid that starts all proteins. He is the starter for every single protein of all the thousands made by your body. Methionine. Methyl 9. He always is the first one on the messenger too. He's the first one. Because that's going to tell the ribosome we're about to make something. Now, he may or may not stay on that protein, but he's going to start it. Okay? Now, this is some ideas. Okay? This is a subunit from the side and from dead on. From the side and dead on. And looking side to side or him on top. And they have, I don't worry about the names. I mean, obviously, you see the head and the neck, or the base rather. The platform, and there's the cleft, like a cleft chin. And you got the stalk sticking up. Stalk makes perfect sense. A protuberance is a projection. Well, since this is the head, we call that the protuberance. Like a head to me. This is a valley. This is also another valley. This is the ridge. Apparently, the ridge is not like a finger like this stalk is, but the messenger goes right through here between these two. The messenger lays on, and that then clamps on top, and the messenger feeds through it. You'll see more about it in a minute. All right. Now, AUG is the code for methionine. That is the code that we've discovered. So if the startup codon is AUG, then on the DNA it used to be TAC, because TAC transcribes to that which is AUG. And then when it comes in, the transfer it brings to him is actually the one called UA. See, I'm reversing it. I'm just reversing it. Now, there are 20 amino acids. There's alanine and all kinds. This is called MET. And like I said, this one does not affect the kind of protein you have. It just starts. It might be cut off later on and not be there at all on the protein. But it always starts. It says right here, although MET is the very first one, it's not always the first amino acid. If I cut that sucker away, I got the second one becomes the first one. You follow me? If I am MET and you're ALA, and they remove me, then you become the first one. Now, when we got made, I was first. But when we leave the ribosome, and I've done my job, I've started, they'll cut me free. And then you become the first one of the protein we're finished. And like I said, it's the second amino acid that determines if I stay or not. It's the second one that determines if I get to stay there or not.
So AUG is the startup.
AUG is always going to be this one here. It's always startup. That's the startup amino acid that starts the show.
For a new one.
For a new one. Now, if this is followed by alanine, if alanine comes second, then I am removed. That's why I said ALA. So when we make this bad boy, it might be 1,000 amino acids long. We're discussing the first two in the series. The first one is always going to be MET. It's going to be MET. Now, if the second one is alanine, ALA, they will cut me free. She becomes the first one then from that point on. I've done my job. I've started it. That protein's finished. They don't need me anymore. So if the second one's alanine, I am released from the protein. I get to start another one somewhere. Okay? So if you see alanine as being number 2, it will become number 1. It will kick methionine out of the way. Okay? Now, lysine is another thing. It's called lys, L-Y-S. If lysine is the second one, then I get to stay. I remain number 1, and lysine remains number 2. And that's how you start the hormone, the protein. There might be other ones, but like I said, the ones we've figured out so far, if ALA is your second one in line, then MET gets released, and ALA becomes top dog. If lysine, LYS, if lys is the second one in line, then the MET stays, and ALA and lysine stays number 2. So if you're lysine, I get to lead the way. If you're alanine, I'm going to be chunked out the side. Now, once we started it, and either I'm there or gone, then that large subunit binds to this complex and starts reading the messenger. Now, the initiation factors, when that large one binds, they're done their job. They bound it to the small unit. See, first thing, you find the small unit and you attach yourself. There were these IF1, IF2, IF3, and then I am always the first one coming in. I'm MET. Now, once that happens, the large unit clamps on, because the large unit receives the transfer RNAs. And I got a good video showing you that. When I went online and looking for these things, I saw some large units, they had 2 sites, but 3 is the one that I understood to be the fact. You have a site for the incoming, a site for the actual bonding of the peptide, and a site to leave. Some folks say a site for incoming and a site for bonding. It just falls off. But I'm going to teach it 3 sites because everything I read is 3 sites. There's your 3 sites. Now this is a large unit. Kind of looks like a boxing glove to me, doesn't it? That looks like a boxing glove with no fingers. Anyway, there's your small, there's your large. This is where your messenger lies. It's going this way. It's going to this base definitely. Here's your sites. All right. A site. Do you remember when I tell you that this thing that's called acetyl? That's the first word, acetyl transfer. Well, the A means first point of entry for the acetyl tRNA. I wish that was called T site, but it's not. The P site is where you actually glue one amino acid to the one that got there previous. And then when the transfer is empty and falls to the exit site, he leaves behind his protein, his amino acid. Okay, so right now some pictures have just these sites, and this one they don't include. But everything I read, this site is still a valid site. Now, this is your amino acid site. It also means acetyl transfer. I prefer to associate A with amino acid, because that's where it first comes in. That's the first entry point in the amino acid site. Now, down here, if this says CCC, guess what the incoming one had got to be? What's opposite CCC?
GGC.
It better be GGG, because that's what the DNA told him. The DNA said GGG. He said CCC. And what's coming in is GGG, which is what I wanted.
It's going this way.
Yeah, we're going to build it this way. He enters, he leaves. He enters, he leaves. He enters carrying, he being the transfer RNA. Transfer enters with the acid attached to him. He comes here, hands off the acid, gets rid of it, and he comes here and he leaves empty-handed to go back out and pick up another acid. This corresponds to GGG.
So that's just like an exit.
It is exit. It means exit. Yeah, it does mean exit. That makes perfect sense. And I know what happens. See, some folks are so concerned about these two sites, they think, surely, it's exit somewhere. But this picture gives you a place for the exit. And you'll soon see what I'm talking about. This is the peptide site. And this is where the actual protein is going to be put together by gluing one acid to a next acid to a next acid. The acids come in this side, get glued together here. The acid carrier transfer leaves, and the acids come out the top, and the chain is the polypeptide, which is protein. Okay. Some books say that the messenger slips across. Some books say he slides up it. My thought is this thing is not moving. This is probably sliding across. It doesn't make a lot of difference in that. If he stays still, he moves that way, that'll work. If he stays still, this slides across, that'll work. So you're going to see videos showing you both descriptions. And you can make your own mind up when you get done watching it. But the polypeptide chain that's going to come out the top of it, that's the protein you're making. That's the protein. Now there's your exit site. At this point, the transfer, he gave it away. He's lost his acid. It's not there anymore. It just falls out and goes out in the cytoplasm and finds another acid to grab hold to so he can bring it back and do it again. But I tell you what, though, now, if this acid is trimine, he'll get more. He'll only get do trimine. He won't get some of the acid because his three bases only correspond to that one kind. He'll pick up more like that one. He wants to. Well, I had alanine next time. I think I'll go and get trimine this time. Same time, same one every time. Probably a boring job, really. But the code he's wearing at the bottom determines you must bring this acid because that's what you're called for, not something else you want to bring. Now, there's that word right there, the A, the amino acyl. And a lot of folks think that this A, and it could stand for that. Some folks stands for the acceptor site. Some say the amino site. I learned that. That's the first. The amino acyl tRNA comes in there first. He's still got his protein. He's still got his acid hooked to him. Comes there, hands his acid off. He leaves empty-handed to get some more acid. Now, this thing is a constant coming in. It's not. They're in line. It's like going through a turnstile to get to a basketball game. It's just a continuous flow. How you give your ticket away, and you next one, you walk up there and give your ticket. It's just like that. And the ticket you give away, that's the amino acid that's being built. And it's not like, one comes in, half an hour later. No, it's continuous. It's real fast. Okay? So, when the first transfer moves to the P site, then the A site can now receive another one. They'll be there side by side. And next thing you know, there'll be three of them side by side as they move through the ribosome. And here's the two I want to show you. The exit site is where the empty transfer just falls away. And that transfer goes back outside the cytoplasm to get another acid to come back and do it again. Let me show you these two YouTubes. These are pretty nice. I always tend to criticize them. I like it to be good for y'all. Let me show you what these things look like. And you get a pretty good idea when you see it, unless they took the sucker off.
When you said that
A transfer RNA molecule is composed of about 75 nucleotides. Each transfer RNA molecule recognizes only one amino acid, which becomes bonded to the top of the transfer RNA molecule. Located on the bottom of the transfer RNA molecule are three